Coagulation on negatively charged surfaces. The effects of vitamin K. Welcome to Chalk Talk. In our previous episode, we looked at coagulation in vivo and presented the cell-based model of coagulation. As seen, a central element of this model is that clotting factors don't just interact with each other loosely in plasma, but mostly bind to the cell surface. This is clinically relevant because this mechanism can be selectively inhibited with vitamin K antagonists to reduce blood coagulability. In this Chalk Talk episode, we'll be taking a closer look at how this works. Let's begin with the binding of clotting factors to the platelet surface. Calcium is a decisive mediator in the binding of clotting factors to the platelet membrane. In contrast to all the other clotting factors, calcium isn't a protein, but is present in blood as a doubly charged positive ion. Using this positive charge, calcium can adhere to the negative surface of activated platelets. In addition, several clotting factors also have negative charges on their surface, which are capable of binding to calcium. So why is this favorable for the coagulation process? Let's take a quick look. Most clotting factors are proteases. That is, they activate downstream factors by cleaving specific segments of the molecule. This is only possible if the proteases and their substrates meet. Compared to the many options for interaction in three-dimensional space, such as in a blood vessel, these options are reduced in two-dimensional space, such as on a surface. This dimensionality reduction is achieved via calcium-mediated binding of clotting factors to the platelet membrane. As a result, clotting factors can be rapidly activated and push coagulation forward. So, as you can see, calcium is an important mediator of coagulation. It's so important that it's even designated as a clotting factor, namely factor 4. To understand when calcium binds to platelets, let's take a look at the platelet surface. As seen in the second Chalk Talk episode on primary hemostasis, platelets change shape as a result of platelet activation, as schematically shown here. This activation causes changes in the molecular structure of the platelet membrane, which consists of a lipid bilayer. In an inactivated state, the outer layer mainly contains uncharged lipids, whereas negatively charged lipids are present in the inner layer. Upon platelet activation, an increasing number of negatively charged lipids are transferred from the inner to the outer layer of the plasma membrane. This results in the creation of a negative charge on the outer platelet surface, which calcium is able to bind to. This transverse diffusion of some membrane lipids from one side of the membrane to the other is called the flip-flop mechanism. The other negative charge that binds to calcium is present on the surface of the clotting factors 10, 9, 7, and 2. Why not try remembering this combination as a year? 1972, the year that saw the beginning of the Watergate scandal. An important element for further understanding concerns the origin of the negative charge of these 4 clotting factors. The negative charge originates from carboxyl groups. The general chemical formula of a carboxyl group is COOH. In blood, which has a pH of 7.4, carboxyl groups are deprotonated. In other words, a proton is released, resulting in a negatively charged COO- group. Things are now going to become a bit more complicated. We'd like to answer 2 important questions. Where are these carboxyl groups located, and how do they attach to clotting factors? The carboxyl groups are located on the glutamate side chains of clotting factors. The correct chemical term for these groups is gamma-carboxyglutamic acid, which implies that it has a glutamate side chain with an additional carboxyl group at the gamma carbon atom of the side chain. So, on to the second question. How do the additional carboxyl groups attach to the clotting factors? This carboxyl group is only attached to the protein after translation. The process is, therefore, termed post-translational modification. So why does all of this matter? This post-translational modification is only made possible through the action of vitamin K, which means that blood coagulability can be positively or negatively influenced by its effect. The reason for this is that in the absence of the gamma-carboxyl group, clotting factors cannot bind well to membrane lipids via calcium. This importance is also reflected in the name vitamin K, referring to its essential role in coagulation. In 1929, the Danish scientist Henrik Dam fed chickens on a vitamin K-free diet. The chickens subsequently developed bleeding. The results were published in a German journal, and the substance was named Coagulationsvitamin, or vitamin K. In 1943, Henrik Damm received the Nobel Prize in Medicine for his discovery. From this important discovery, drugs were later developed that are now vitally important in daily clinical practice. These are the vitamin K antagonists such as warfarin. They prevent the carboxylation of clotting factors and, therefore, work precisely on the mechanism previously described. In the next slide, we'll be taking a closer look at vitamin K-dependent clotting factors. There are 4 clotting factors that use gamma-carboxyl groups to bind to the cell membrane. As previously mentioned, these factors are 10, 9, 7, and 2. In addition to these 4 factors, there are other vitamin K-dependent proteins. Examples include proteins C and S. Both of these proteins are inhibitors of coagulation. Okay, let's now take a look at the positions of factors 10, 9, 7, and 2 during coagulation. At this stage, it would be beneficial to have watched the previous Chalk Talk episode on the cell-based model of coagulation. But don't worry if you haven't, here's the link. Factor 7 is part of the extrinsic tenase complex, which is formed at the beginning of coagulation. Factor 9 is part of the intrinsic tenase complex, whereas factor 10 is part of prothrombinase. Therefore, all 3 factors are components of complexes that play a central role in coagulation. The 4th and final factor, prothrombin, is also bound to the membrane. This is quite useful because prothrombin is directly delivered to prothrombinase. Prothrombinase is a complex comprising activated factors 10 and 5, which is also membrane-bound. This interaction results in rapid activation of prothrombin on the cell surface. By the way, recent studies have shown that other factors are also capable of binding to the platelet membrane, such as factors 5 and 8. However, the exact mechanism by which these factors bind is still not completely understood, but is under investigation. Okay, in this episode, we've shown you how clotting factors bind to the cell membrane. In some textbooks, you may find the following alternative notation for the complexes formed during the process. This notation may be initially quite puzzling, but it should be easier to understand at the end of this episode. These are complexes of activated clotting factors, in this example, of the factors 10 and 5. The second part indicates that the complex is bound to the phospholipids in the cell membrane by calcium. The older term platelet factor 3 is sometimes used for phospholipids. The term was established after discovering the contribution of platelets to the coagulation process. Today, we know that platelet factor 3 is the membrane of activated platelets, more precisely, the negatively charged phospholipids that are exposed through the flip-flop mechanism. So that just about wraps it up for this Chalk Talk episode on secondary hemostasis.